In this video, we will discuss about this aberrant glycosylation on cancer progression. Though we have covered this topic many times in our previous videos, in this video we will cover something new. We will try to cover something new or try to explain the things in a way that normal people will be able to understand these things. So we will start with this O-glycnation of transcription factors. So what is this O-glycnation of transcription factors and how it will affect this cancer progression. So this O-glycnation is it means that when this glycanac is added to the any substrate, so then it is known as O-glycnylation, and this OGT helps in the formation of O-glycnylation, and this OGA removes this glycanac, so it removes this O-glycnylation. So this is the cycle that occurs. And what happens in the case of cancer, this O-glycnylation of several transcription factors occurs, and this O-glycnylation uh, helps the transcription factor for this nuclear localization, or this helps in transferring this helps in the movement of transcription factor within the nucleus that we can see here. And this transcription factor will then interact with the DNA and upregulate the proteins that are responsible for anti-apoptotic response, and also promotes the proliferation of the cells. So in other terms, we can consider it as this O-glycnylation promotes the anti-apoptotic factor, that is, it prevents the tumor cells from death, from triggering this programmed cell death because of the change in the biological pathway. At the same time, it will also promote the proliferation, or in other terms, it will promote the uncontrolled growth rate of these tumor cells. So this is how this O-glycnylation works. However, this O-glycnylation also affected by some external factors that are shown here. That is this new one, RTK, FBL3, and MUC6, MUC4, HERT. And so what this transcription factor does is a transcription factor regulate this OGT and OGA amount, and thereby this O-glycnylation will get regulated in the case of this when this receptors get activated. And one more thing, this receptors when get activated, it will cause a hypoxia condition, or we can say this uh, a stress condition where there is a scarcity of the oxygen, and that will trigger more formation of this O-glycnylation, and thereby helps the tumor cells uh, from the death by triggering anti-apoptotic factor, and at the same time it will promote uncontrolled growth rate by upregulating this proliferation related proteins. So this is how O-glycnylation works towards cancer progression. Now let us understand how this aberrant glycosylation occurs in the case of pancreatic progression. So this is the normal pancreatic pancreas. Here we can say the normal glycosylation profile. However, with the increase in mutation, or with the movement of this pancreas towards this normal pancreas towards the pancreatic pancreas, we can see that this sialic acid formation has been will increase, and will further progress will further progression of this cancer. We can see this truncated O-glycans, such as this TN, STN antigen will increase, and thereafter this mucin formation will also increase, that we can see here. This S. levis AX carbohydrate antigen will starts to form, and finally when this glycan will be able to interact with the uh, tumor microenvironment, or the environment near the in environment the neighboring of the tumor cells, and when they reach at this stage, then the metastasis process begins, that is this extravasation of this primary tumor into the blood vessel starts, and after that when it reaches close to the secondary organ, then this with the help of selectins such as E-selectins, P-selectins. The intravasation of these tumor cells occurs, and then it will grow in those secondary organ, or spread those tumors in the secondary organ. And this is the big picture that is shown here. That is, metastasis begins at the liver, at the pancreas, and then it will goes into the lungs and spread the tumors on the lungs. So this is in brief how this O-GlcNAcylation works towards cancer progression, and how the other O-GlcNAcylation process changes as the normal pancreas will move towards the pancreatic pancreas, and how the metastasis occurs in brief. If you need to understand more about this cancer progression and glycosylation, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.